Okay, so today and tomorrow we're going to focus on the last chapter that we cover in ANP1. And this chapter, we've already snuck some of it in because it's kind of hard to talk about the nervous system without talking about the senses to some degree or another. So some of what we do, at least initially, will be a little bit of review, but a lot of it's going to be brand new stuff. And it's going to be just sort of systematically looking at senses and how senses are talked about in anatomy and physiology. Well, it should make sense to you that most of your senses give you awareness of your external environment. Am I right? So when you think about the things you're conscious of, the sensations you're conscious of, you've got touch and vision and hearing and taste and smell, and all of those are ways that we sample the external environment. In fact, of all the senses that we've focused on this semester, there's only one that isn't an internal sense. And we'll come to that in just a second. So your sensory information gives you internal and external information. It has to come in from receptors, and we're going to classify our receptors. So the first thing I want you to do in your groups, list all the types of sensation you can think of. Let's spend about 30 seconds on this. It shouldn't take you very long. Well, maybe more than 30, but not very long. When you were in 5th grade, everything happened in 5th grade, right? That's when you learn your Roman numerals. When you're in 5th grade, you learn the 5 senses, right? So what are the 5 senses? Touch, smell, taste, hearing, vision. So those I know you'll get, but is that all there are? Beyond the basic 5. Beyond the basic 5. Oh yeah, put ESP down. That's a type of sensation. No imagination. What do you have? Pressure and temperature. How about that? Pressure. What do you guys have? Something else? Pain. Is that a sense? Is the basic 5? Come on. All the types of sensations. Yeah, I don't think of anything else. What? Okay, now, having done that, and not seeing what I'm looking for on anybody's list, what are you looking for? Taste, we got, that's one of the big 5, right? What were the big 5 again? Taste, touch, smell, hearing. Emotions are not sensations. No. You can feel them, yes, but they're not based on sensory receptors. No.
I wrote light.
Okay, that's vision, though. Okay. Yeah. Okay, so you did the eye and ear lab when? Thursday? Okay, so you learned the parts of the ear, right? Was every part of the ear involved in hearing? No. So what else was there besides hearing? Equilibrium or balance? Same thing, more or less. Is that a sensation? Yes. Yes, it is. Okay, and none of y'all put it on your list even though you all studied the ear just last week. Okay? Your sense of balance is actually the fifth of what we call the special senses. So we're going to talk about the difference between the special senses and the general senses as we get into our kinds of receptors. Okay? So what does it take to be a sensation? Sensations have to be picked up by receptors. And I'm not talking about overnight sensations, not that kind of sensation, okay? Sensation as in sensory input to the central nervous system. Some of them are simple, some are complex structures. These are the ones we call the sense organs. And we're going to classify our receptors. We're going to be spending some time on that. So one of the ways we classify our receptors is it simple or is it complex structurally? Okay, another way we classify them is by the type of sensory information, and this depends on the stimulus. So we have photoreceptors in our eye, right? They respond to photons of light. We have mechanoreceptors in our ear. They respond to sound waves, pressure waves. All right, so it depends on what kind of stimulus that receptor responds to. And just as a thought experiment, we would never actually do this in real life, but imagine if you could take the optic nerve and the cochlear nerve. Where does the cochlear nerve come from? From the cochlea, okay, for hearing, right? So if you could surgically in an experimental animal cross the cochlear nerve and the optic nerve, just slice them and then reconnect them to each other so that the cochlear input went to where the optic nerve's destination is, which is visual cortex, and vice versa, then you would see sound and hear sight. Because the interpretation by the brain is wired to particular sensory receptors. So just let that idea kind of gel for a minute. So the stimuli that are picked up by the receptors are like tuned to a particular kind of stimulus, and that is just another way we classify our receptors. So our types of sensation, we have really three kinds, sort of, kind of. Two of them sort of go together. So conscious awareness, you're all familiar with that. You can figure out what that is. Subconscious is not necessarily the same thing as visceral, but let me try to make a distinction. Visceral are subconscious. You're not conscious of them. But visceral sensations are like those things that are the receptors in your bloodstream that are responding to pH changes in your blood, or the receptors in your stomach that are responding to the amount of fat content in the food you just swallowed. Those are definitely visceral. Is there any way you could turn your attention to them if you wanted? Can you sit there and think, I wonder if my pyloric valve is open or closed? Any way you can direct your attention to that? No way, right? But if I asked you to turn your attention to subconscious things, then to some degree you can. So for example, what's the noise level in this room if we're all quiet? What do you hear?
Air conditioner.
You hear the air conditioner. Did you hear it before I pointed it out to you? Yeah, okay. How about the room temperature? Warmish today? Not too cold? Definitely not. How about the feeling of the neckline of your shirt? Is that bothering you? I have one shirt that's got some little sharp thing that sticks me. I finally quit wearing that shirt. But unless it's really bothering you, you just tend to tune that stuff out. Okay, so you get used to things. You adapt, we say. So those, to me, are truly subconscious. And a lot of your subconscious things, you have some conscious awareness of, but you don't have to. So you have to pay attention now to the contraction of the muscles, the erector spinae muscles in your back that are keeping your back upright. Paying any attention to those right now. Yeah, so that's kind of, to me, subconscious. You could be conscious of it, but most of the time you just filter it out because you don't need to pay attention to it. Okay, so we have these lower areas of the brain that are getting that information, and a lot of it's either getting relayed to parts of the brain that don't talk to your cerebral cortex, which you now know is the seat of consciousness, or they just tend not to. And so you remember from last week that the thalamus is your sensory filter for everything except your sense of smell, right? So if you're sitting there and you're reasonably comfortable and your clothes are fitting you okay and the room's not too warm and the air conditioner's not too loud, you're able to just tune all that out and focus on studying for your test or whatever it is you're really doing while you're sitting there. Okay? So the thalamus filters all that out. But if it's your heart rate and your respiratory rate and the state of the chemistry of your bloodstream, that doesn't even get to the thalamus. It's going to the hypothalamus. The hypothalamus, remember, is the master regulator of your visceral sensation. So you're not aware of that at all. All right. So for a stimulus to be consciously perceived, what area of the brain has to receive it? You're going to be conscious of it. What area of the brain has to perceive it? Are any of y'all relying on that monitor? Anybody? That one's blinking in and out. It's making me insane because I am ADD, so it bothers me. I'm just going to turn that on. All right, so that doesn't take long to answer. What's the answer to that? Who's brave enough to tell me what the answer is? Conscious perception? What are your choices? What parts of the brain might it be? Could be the thalamus, that's your sensory filter. Could it be the hypothalamus? What did I just say about the hypothalamus? Visceral. Okay, not conscious, visceral. So if it's going to get to the thalamus, where does it have to go from there? Sensory relay to what? You're having a test on that today, you guys. So where's all your conscious perception? That's probably too specific. Yeah. So cerebrum is too general, frontal is too specific. It's actually slightly off. How about just cerebral cortex? It's what we call a cortical function, okay? And if you mapped it correctly, you would know frontal is motor, right? Consciousness is the back of the brain. So conscious perception is a sensory awareness, and it's going to be in parietal lobes for tactile, right? And for taste, also parietal lobe. For hearing, it's temporal. For vision, it's occipital. You knew that, didn't you? Okay, so I'm just asking you to pull all that together under one big term. Cerebral hemisphere, cerebral cortex. Cerebral hemisphere actually includes the white matter and all the other parts. So just the cortex and just the back part behind that central sulcus. Behind that is where you're going to have sensory perception. All right, so here's something that's probably new to you guys, the idea of a transducer. Now, if you played with electric motors when you were a kid, then you may be familiar with the term. So any of you play with toy trains that actually ran on a track? Not little wooden ones, but one with an engine that turned? Play with those? Do you remember what we call the little power box that you turn on and off? That's the transducer. A transducer is something that converts energy from one form to a different form. And so we use that term in engineering, but we also use it in physiology. Transducers in your body are your receptors. The receptors take energy in one form, and they convert it to a different form. So transduction is the conversion of that energy. When light rays hit your retina, they get converted to nerve impulses. So we change the energy from light energy to electrical energy. When sound waves hit your ear, they get transmitted to the cochlea, and in the cochlea, they get converted to nerve impulses. So again, we're converting mechanical energy to electrical energy. Every single sensory receptor converts its stimulus input into the electrical energy of a nerve impulse. So that's what transduction is. And so you need to know what the term means and realize that's all it means, okay? So when I tell you something's being transduced, just in your heads, translate that into, oh, I'm just converting stimulus input into nerve impulse. That's all that means. Okay, next concept, the receptive field. The receptive field is simply the distribution of nerve endings, wherever they are, into an area of the body, okay? So some receptive fields are quite small and some are quite large, and we're going to kind of practice with this as a little bit as we get into the different senses. And we're going to start with the skin. So what I want you to do is work with a partner, and don't work with a body part that might end up getting slapped across the face. Be careful. But what you need is two sharp objects, 2 pinpoints or something, pencil, something sharp. And what you want to do is practice with your partner's eyes closed so they can't see what you're doing. You're either going to hit them with 1 pinpoint or 2, and they have to tell you whether it was 1 or 2. Okay, now, before you move on, you're going to try different parts of the body. I suggest fingertip, arm, and maybe back of the neck or something like that. Again, being careful not to invade someone's private space. And you have to move the 2 points closer together. If they can still tell it's 2, move them closer together and see if they can still tell. But alternate 1 point and 2, 1 and 2, until you get some sense of mapping their receptive fields. And so on the diagrams on the screen, you can tell the difference between a small field and a large field. Okay, let's spend about 1.5 minutes doing this. So work with at least one partner. If you want to work as a table, that's also okay. with poor equipment and no training. So if you didn't get the right answers, don't feel bad. But let's take a look at our picture and see if we can make some sense of what should have been revealed. So here we're seeing small receptive field in the fingers, large receptive field on the skin of the back. And what that means is that the nerve supply to the fingers is denser. In other words, I have more nerve endings in the skin of the fingers than I have in the skin of the back. Okay? And by having more nerve endings there, then what that means is that if I stick a pencil point here and another one here, I'm stimulating different nerve endings, and therefore I'm going to feel it separately. Whereas if I took that same distance on the skin of the back, so the 2 points same distance apart, but the skin being hit is in only one receptive field, so I only feel it as 1 point. Okay? So a large receptive field means a larger area of skin supplied by one nerve ending. A small receptive field means a small area of skin supplied by one nerve ending, which means I have more nerve endings. So I have more receptive fields in the parts of my body where the skin is more sensitive. And generally speaking, for most of us, that's our fingertips, because that's how we explore our environment, typically. Make sense so far? Questions about that? So how many of you got results that are consistent with our expectation, which is that your skin was, your fingertips were better able to distinguish 2 points close together? Yeah? Okay. So some yes and some no, and I don't even begin to have an explanation of that. All right, so then here's your next question to answer. Which has a larger area of the cerebral cortex dedicated to its sensation? The fingertip, which is smaller receptive areas, or the equivalent area of skin from the back, which has larger receptive areas? Okay, write your answers down on your, discuss them in your groups and write them down. Okay, so looking again at the picture we just looked at, which one has more nerve endings? Fingers, okay? So does that help you answer the question? Which one has a larger area of cerebral cortex for the same amount of skin? The one that has more nerve endings. And reminding ourselves of this picture that we just looked at last week, if we find the skin of the left, or of just the index finger, that is right here. That's the entire index finger. And then the skin of the back, the entire trunk, is right here. That's the entire trunk. So all of the skin on the entire trunk. So how much of your cerebral cortex is devoted just to your index fingers compared to the entire skin of your trunk? It's a huge amount, right? So you have lots more nerve endings, which means more sensory receptors in your fingers than you have in your back. And of course, what other part of the body also has lots of sensory receptors? What parts look unusually large in this diagram? The hand, the face, the tongue. Okay? Makes sense that those places would have more sensory receptors. Marvelous. Okay, so now we're moving on to another aspect, another quality of receptors, and this is how long does it send the stimulus? And there are two choices, the first of which is tonic. And tonic is the same word root as tone. So we've looked at tone. We looked at muscle tone. We looked at autonomic tone, which was sympathetic or parasympathetic. With sympathetic, we learned vasomotor tone is continual stimulation of your blood vessels, whereas vagal tone is continual slowdown of your heart. So tone should mean something to you now. It should mean chronic, continual impulse conduction. Okay? So if I have tonic receptors, tell me what they're doing. They are tonically sending signals. That is, they are continuing to send signals even if the stimulus that started it is gone or hasn't moved. Okay, so tonic means you send a signal for a long time even if the original stimulus is gone. Best example I can give you to this is pain is a tonic sensation. So last time you burned yourself, think back, how quickly did you remove the hot object from your skin or your skin from the hot object? Instantaneously. Did it stop hurting instantaneously? No, it continues to hurt. That's the idea of tonic sensation. Tonic means I keep sending impulses even though the stimulus may long be gone. Continues to respond to the stimuli. So another example, the one on the screen, balance receptors in your inner ear. So you've quit moving your head, you're sitting rock steady. So there's no new stimuli, right? But your ear receptors don't quit sending information. If your ear receptors quit sending information, then you feel like you're floating. You ever had that sensation? Okay, it's really weird. Okay, we'll talk some more about that. So basically, they don't adapt. Adaptation is getting used to things. So our tonic receptors don't adapt, or they adapt very slowly. Okay, they don't get used to it. They just keep saying, hey, there's something going on, even if the original stimulus is gone. All right, that's tonic. The opposite is a phasic receptor. So think of phases as being rapidly changing scenarios. And phasic receptors are really, really, really good at responding to rapid changes in whatever stimulus that receptor is designed to take advantage of. So tactile stimulation, because tactile stimulation, if you've got a bug crawling on your arm, you need to be able to respond quickly. You don't want to think it's back on your hand when in fact it's made it all the way up to your neck. So you don't want tonic for that. You want phasic. You want it to adapt quickly. Okay? So what happens with phasic, though, is that you reduce sensitivity. So if stimulus arrives and then doesn't change, then these phasic guys quit sending signals. Okay? And we're going to look at some examples of those, and I can't remember. Let's see. So let's work on these three. Your awareness of your body position, temperature, and water that you're immersed in, and sensation of pain we already did. So that should give you a touchstone. So for the first two, tell me whether you think they're tonic or phasic. In your groups, writing down the answers. So all I have to do is tell me if it's tonic or phasic. Tonic or phasic. Okay, so for this, which one adapts? Do you remember what adaptation meant? What was adaptation? I just gave you that. Getting used to it. So do you get used to pain? Do you get used to pain, or does it continue to send a signal? So did we say that was tonic or phasic? Tonic, you keep sending signals. Do you get used to the temperature of your pool or bath water? Do you get used to it? Yes. So is that tonic or phasic? Okay, I'm hearing wrong answers, so that always makes me stop. Do you get used to the pain of, in other words, does it quit sending pain signals? No, it keeps hurting. You didn't get used to the fact that you damaged those cells. It's still sending those signals, right? Pain lingers. It does not get used to it. That's tonic. So you do get used to the temperature of pool water or bath water, right? So it's phasic. If you get used to it, it's phasic. In other words, if you quit feeling it, okay? So if you went swimming, anybody get to go swimming over the 4th of July weekend? Anybody? None of you guys did you? Was the water cold when you got in? Felt good? Cool and refreshing? Cool and refreshing is good. My pool's already too hot. But you've been in pools that were too cold when you got in them, right? Yeah. So what's your preferred mode of entry? Do you just jump in all at once? Yeah, my husband does that. I cannot make myself do that. If it's cold, forget it. I inch in. But you get used to it as you go, right? Or you cannonball in and you get used to it. You get used to water temperature. That's phasic. If you can remember that as your example, it'll help you keep them straight. Yes?
On here, it has tonic as getting used to.
It has the word tonic. It introduces it. Okay? But that's not what the meaning is. All right, it's on. Maybe I need to move that to the other screen to be less confusing. But that's part of the point of doing a lecture, is to help make sure we explicate it. Okay? You would all agree that in common usage, you get used to bath water, right? So you got in the bath, it was nice and hot, and then it cooled off, didn't it? And you had to add more hot water. It didn't cool off. It doesn't cool off that fast. You just got used to it. So you add more hot water. It's the change in temperature that your phasic receptors are responding to. They respond to change. Phasic, change, tonic, tone, steady state. Okay? So what about your awareness of body position then? Phasic or tonic? Do you lose your sense of awareness of your body position the way you lose your sense of the pool water's cold or the bath water's warm? No, you really don't. You don't. Let me explain that one to you. That one's tonic. Because once I explain it, you'll either go, yeah, I've done that, or okay, I've never done that, but I believe you. So here's the setup. You wake up in the middle of the night, you're so tired you went to bed and you didn't move. For 6 hours you didn't move. And when you woke up, you couldn't feel your arm or your leg. How many of you has that happened to? Look at the hands in the room. That's like almost half. So for those of you that didn't raise your hand, do you believe that? Can you trust that this many people say, I woke up and I couldn't feel my arm? Anybody want to give us a testimonial? Anybody?
It'll happen after this class, though.
Yeah, no doubt. Thursday night, y'all are all going to sleep solid. And when you wake up, you're not going to feel your arm. So what happens if you can't feel your arm? It's like, I'm telling you, it's like it's not there. You can see it. As soon as you touch it, it's back. Okay? But as long as you're just laying there not moving, it's not a sense of my arm's floating in space. You don't have an arm. Your brain says, what arm are you talking about? And even though you can see it and you know it's there, you can't feel it. Yeah.
Mine will start hurting after.
That's tingling.
No, I mean
Like hurting? Oh, your brain's different than most people. That's a nice way of saying you're atypical. Not abnormal, just atypical. Okay, but for most of us that have had this sensation, you're laying there and you're thinking, all I have to do is move it. It'll come back. Okay, can you move it? You're not moving anything else. It happens so rarely, the chances of it actually happening on purpose when you're going, oh boy, I finally get to experience this, not happening. But imagine, if you will, that you're laying there. If I can just move my arm, then it'll pop back into my consciousness, right? Except your brain is saying, what? I don't know what arm you're talking about. You want me to move what? There's nothing there. So you have proprioceptors. Remember what those are? Your sense of awareness of your body part. They are tonic. They are continuing to send impulses. You have to lay really still for a really long period of time before they finally quit sending signals. Okay? And so consequently, this happens extremely rarely. All you have to do is grab it with your other arm, the arm that you do know where it is. As soon as you move it, your brain goes, oh, that arm. And you're back. Now, this happened to my sister. Both her arms went to sleep, but no sensation at all. So she was laying there. She'd done this before. Heavy sleeper, apparently. I don't know how she does that. We used to have to share a double bed, so I can't imagine that I laid still enough for her to lay that still, but there you go. So she finally figured out, because she couldn't move her arms at all, that if she just tried to sit up, that would move them, and that worked. And her arms popped back. So in case you ever need a scenario. There, you've got your solution. But it does happen. Now, if you can just remember that, then you can remember that that's what we mean by tonic. So tonic means it keeps sending signals even though the last time you stimulated it may have been 6 hours ago. It still sends signals. Okay? That's tonic. All right, so let me ask you a different one. What about your sense of smell? Sense of smell.
I would say phasic.
Phasic, and you'd be right. Okay, so sense of smell wears off rapidly. And there's a good reason for that, and there's two different ways we get used to things. But with sense of smell, it actually uses both, to my understanding. But you get used to smells, right? You do. You get used to them. Some smells you never get used to. But back on an earlier slide, I was showing you some things alert you to danger, and one of the things that alerts you to danger is there are certain smells that automatically trigger danger scenarios, and one of them is the smell of skunk. You imagine that sends danger signals to your brain. And your sense of skunk awareness is very finely tuned. It doesn't take a lot of that chemical for you to be able to sense it. And that's actually what we add to natural gas. You know, natural gas itself is colorless and odorless. So if you have a natural gas leak in your apartment and you have all the windows closed up, what's the only thing that makes you alert to the fact that you have a gas leak? You smell it, but you're not smelling the gas, you're smelling the mercaptan, which is the skunk odor that they add to it just to make sure that you can tell when there's a leak. The problem is that because it adapts fairly rapidly, that if it was a slow leak and you were sitting there or sleeping there or whatever, then it could build up to dangerous proportions, and your sense of smell adapts so rapidly that you might not even notice it, even though someone else coming into the room would just be knocked back by the odor. You got used to it that fast. Okay, so sense of smell adapts very quickly. It's phasic. Okay, questions about the difference between tonic and phasic? Got those pretty straight? You have a list in your online materials to make sure you will. All right, so we've gone through these classifications, and now we're going or we've gone through the characteristics. Now we're going to classify receptors. Somatic and visceral, we already know what that means, right? Somatic means of the body wall. Visceral means of the internal organs. The location is either exteroceptors, visceroceptors, or proprioceptors. Do we remember what proprioceptors are? What are they receptive to? Proprioception is awareness of the position and movement of your body. Okay, so mostly arms and limbs, but also head and trunk. Okay, and then by the complexity, they're either general or special, complexity and distribution. So we're going to take a look at those, and we'll start with just the eye as an example. So can you see our three categories? I'm going to go back to the previous slide, and you're going to classify the eye. Is it somatic or visceral? Go ahead and write them down. Do them in your groups. Is it somatic or visceral? Is it extero-viscero or proprio? And is it general or special? It should not take you long. We're just doing the eye as an example. Is anybody not done yet? Is anybody not started yet? Okay, so what's the first one? Is it somatic or visceral? Somatic is consciousness, visceral is visceral. You can't be conscious of it, right? Are you conscious of the input from your eye? Are you not? Can you see? Okay, can people in general become conscious of the sensations coming along the optic nerves? Yes. That's a simple question. Yes, it's a somatic. Is it an extero, viscero, proprio? Well, where is it? It's on the outside surface. It's responding to stimulation from outside the body. So clearly exteroceptive. And then is it complex or simple? Your eye, you studied it in lab. It's pretty darn complex. The eye and ear are the most complex. That's why we study them in lab. And then where are the receptors found? This is a dumb question. You're in your head, right? Okay. So what I want to do next is let's just talk about the special senses because they are special. But I want you to be aware of what makes them special. So let's list them first. Let's list the receptors, not the senses, because we kind of got that figured out. So we have eyes. What else?
Ears.
Thank you. What else? Nose. That's fine for now. And then tongue for taste, mostly tongue. Okay, can you all agree that all of these are in your head? Okay, so these are all, some are more complex than others, but all of them are in your head. Are there any other special sense organs besides these? We have eyes for vision, right? We have ears for two. What are our two for ears? Hearing and balance. And we have nose for smell and tongue for taste. And that's it. That's our 5 special senses. What makes them special? They're all in the head. They're not all in your head. They're all located. The receptors are located in the head. Do they have to be in the head? Could you have visual sensation from, say, your hands? Would that be kind of cool, wouldn't it? Just hold your hand up. You could see with you. If you had an eyeball right there on your hand, that'd be pretty cool. But no, they're all in the head. Okay? So they're specialized receptors for monitoring the external environment, and they're all located in this very mobile head. Okay? Those are the specials, and they're the only specials. All right, so we got that under control. All right, modality. This is another word that you need to know what it means. The mode is the type of stimulation. So our modalities include mechanoreceptors, thermoreceptors, chemoreceptors, and photoreceptors. Most of those words you're familiar with. And then there's some other subdivisions. So a mechanoreceptor is one that we've met before because we talked about the receptors in the skin. And what we said was you can poke it, shake it, stretch it, do anything physical to it. If it's a physical stimulation, it's a mechanoreceptor. Okay, so can you explain to me why the receptors in your ear are mechanoreceptors? What's that?
Because there's physical movement.
There's physical movement, absolutely. That's a good, so you have the compression of the sound waves coming in, you have the actual pressure waves hitting your eardrum, creating a little domino effect through the inner ear, and it's the physical movement of that fluid that makes those cells send impulses. Likewise for balance, you've got the movement of the head creating changes in fluid. So those are mechanoreceptors. So tactile, remember, is touch receptors. What is a baroreceptor? Baro as in a barometer, which is measuring pressure. So that's a pressure receptor, which in effect is a stretch receptor. How do we measure pressure? How do you suppose your urinary bladder measures pressure? By the stretching of the wall. So pressure receptors really are stretch receptors. Okay, and then another one that should be added to this list but isn't there, of course, is the internal ear receptors. Those are all mechanoreceptors. Thermoreceptors respond to temperature. We don't exactly know how they work. Chemoreceptors respond to chemicals. What is a nociceptor? The word root noci means the same thing as noxious. Same word root as noxious, which means irritant or whatever. Nociceptors are for pain. And then photoreceptors respond to photons of light. Okay, questions about the modalities? All right, so this is a quick review. This was actually on the online lab for skin, the sensory receptors. Y'all don't mind if we go over it today since it's on your lab test tomorrow? Okay, very cool. So the receptor types found in the skin, most of which are also found in the mucous membranes. Some are encapsulated, some are not. You need to know what layer they're in, and you need to know their functional specialties. So we have, this is the picture out of your book, and you've got two categories, unencapsulated and encapsulated. And you have three kinds of unencapsulated: a tactile disc, the free nerve endings, and the root hair plexus. And of those, which one would not be found in a mucous membrane? Yeah, you really don't have hairs in your mucous membranes. Really, really. Hairs are found just on skin. Okay? So a root hair plexus is the one that is responding to stimulation or movement of the hairs. So if a mosquito lands on your arm and it makes the hairs move, or the breeze blows and it makes your hairs move, then those root hair plexuses will respond. Free nerve endings are found in both the dermis and in the epidermis. And in general, we probably are oversimplifying this, but in general, a pain receptor is going to be in the epidermis, and the thermoreceptors are down in the dermis. They're pretty high up in the dermis, but that's just sort. It's probably an oversimplification, but we know that there are pain receptors in the epidermis. How do we know that? We've done that experiment on ourselves, whether we meant to or not. We've all got paper cuts before. Do they hurt, even if they don't bleed? There are pain nerve endings in the epidermis. And thermoreceptors, I've read, are in the dermis. Okay, so free nerve endings. Tactile discs are very specific ones that sit in the stratum basale, and they respond to very light touch. So again, that's something that your mucous membranes are not that involved in, so while they may be found in mucous membranes, they're not going to be exceptional. Our encapsulated guys, we have four. We have tactile corpuscles and bulbs, bulbous corpuscles, and lamellated corpuscles. And I'm sorry, you just got to figure out which one's which. Okay? Tactile corpuscles are up at the highest point. They're up in the dermal papillae. They respond to more continuous touch. And yes, you have them in your mucous membrane. So imagine swallowing too much food in one bite. And you can feel it stretching your esophagus as you swallow it down. You're stimulating these. You're also stimulating the bulbous guys. Look at these bulbous guys. Do you see how they're kind of long and skinny? So what they have is they have little fibers of connective tissue that spread out into the surrounding tissue. So when you stretch that tissue, those guys get stimulated. Okay, and our lamellated guys have lamellae. You learned that term associated with the bone lab. Do you remember that? Remember you had lamellae, which was just layers and layers and layers. So when you look at that, what do you see? Layers and layers and layers. Okay, these are just little layers of like a gel capsule. And these guys, because they're very deep, respond to really deep pressure, like somebody poking you, or vibration, like if you were in an earthquake. Your little lamellated corpuscles would be going nuts. And you have them not only in the deep dermis, you have them in your joint capsules. Actually, they're in the joints. So they're associated with proprioception as well as exteroception. Okay? So that is the quick review of the tactile receptors in the skin. Okay, something much more interesting to talk about is referred pain. So we know that our viscera do not have a road to consciousness, right? So can you tell me right now whether your stomach is contracting or relaxing? You can't feel it. You couldn't feel it even if you tried, okay? You cannot turn your attention to the stomach and feel it. But if you had a stomach upset, would you feel that? Yeah, okay. So here's the deal. Pain is a stimulation that is interpreted differently than somatic stimulation or typical somatic stimulation. So in pain sensation, that gets to the level of the thalamus, and the thalamus says, oh, it hurts. That's different. So if it hurts, wherever the damage is coming from, You have to have a way of knowing what hurts. Where is that pain coming from? And so your brain has this process, we haven't got to it on our list yet, called projection. So let's talk about projection, and then referred pain will make some sense. Okay, so projection is the brain's ability to interpret the source of stimulation. So if you poked yourself in the right index finger with a sharp object, it would hurt. And you would know where it hurt because not only do you have the pain sensations originating from that particular receptive field, but you also look at it and you see blood coming out of it. Okay? So you have other ways of confirming the source of that pain. And you don't remember learning that, but you did. You learned it as a baby. You learned, just like my little 2-year-old is learning now, not to bite his cheek. That's something he's having trouble with. It hurts. And we can tell that's what's bothering him because, first of all, he's learning how to talk, and second of all, he cries in the middle of chewing dinner. So he's sitting there eating chicken nuggets, and he starts crying. What else could it be? But he bit his tongue and bit his cheek. And he'll stick his tongue out. If it's not bleeding, I figure it's the inside of it. I'm not going to look that far back to see. But. So you learn these things, and that feedback is part of the learning process as children become mobile. So the other thing he's having to learn right now is not to hit his head. So we can tell he's learning that because he hits his head periodically, which makes him cry because he's 2. But he's also learning how to look up when he ducks. So he's learning to plan ahead and not hit every countertop in my kitchen. All right, so pain is for learning. Pain teaches you not to do that particular thing again. The problem with referred pain is there's nothing you can do about it, really. So the pain signals get to your brain, and your conscious brain is trying to project those sensations back to the source. And it's going, I don't know what you mean by my heart is having an attack. So the closest thing I have to that is the esophagus or the chest. And so the brain projects those sensations back to the nearest somatic nerve. The nearest somatic nerve, which may not actually be the nearest one in the adult, but it was growing with it in embryonic development. So we localize these stimuli back to the nearest source, and the nearest source may be the skin of the abdomen. So you may have appendicitis. Where does it hurt? Where does appendicitis usually hurt? Lower right quadrant. Okay? Now, in some people, the appendix may be there, but if it grew differently, it may hurt somewhere else. Or it hurts there, but that's not where the appendix is. Depending on your personal anatomy, the appendix may be in the wrong place. This is why we pay the surgeons the big bucks, right? So does everybody understand the idea of referred pain? The nerves sit side by side. The visceral nerve sitting right next to this particular somatic nerve is going out to this dermatome right there. And so referred pain is your brain's projection of a visceral injury to the nearest somatic source, because somatic is the only thing the brain, the conscious brain, knows of. Well, we're able to, for most people, map referred pains. You do not need to memorize the map for the test, but you need to have some sense of the map. So if you were having a gallbladder attack, where would it hurt? Usually, right up under the ribs on the right side, but notice it can hurt up here. Okay, so it just depends. Kidneys, usually you have lower back pain. So if you have lower back pain and it's excruciating enough to go to a doctor, particularly if it's bilateral and not just on one side, then first they're going to quiz you about what have you been doing muscle-wise, lifting heavy objects with your back or whatever. But if that's ruled out, then they may do some kind of imaging of the kidneys to see if there's a problem. Okay? So does everybody understand the idea of referred pain? These maps are idealized, they're generalized, and true to form. They're more typical of men than they are of women. So you see that a heart attack may hurt down the left arm. So we see that in the picture. And that almost never happens in women. In fact, in women, the symptoms of a heart attack are so utterly different from what they are in men. Men do usually report pain, and women usually do not. They report a feeling of tightness or just pressure on the chest, and rarely, if ever, pain down the left arm. Now, I want you to find, it's not in the picture. I'll have to draw it in. The esophagus. Where does your esophagus sit relative to your heart? parts here, esophagus, pretty much right behind it. So guess where your esophagus hurts? So if you have acute esophagitis, an inflammation of the esophagus, then it may hurt in your chest. And so you go into the hospital thinking you have a heart attack. And they'll strap you up to all those heart monitoring devices and do an ECG and take some blood work and say, no, not a heart attack. And so then they start talking about viral esophagitis or what did you eat and do you have reflux disease and so forth. So there's a lot of overlap, and sometimes the patient simply cannot accurately report pain in any way that allows you to nail down a diagnosis. Okay, so does everybody understand referred pain? All right, let's distinguish that from phantom pain. Phantom pain occurs when a body part that you've had removed still feels like it's there. That's phantom sensation. And if it hurts, that's phantom pain. What are you going to do about that? If you had your leg amputated right at the knee and your ankle feels like it's throbbing, how are you going to treat that? Pretty much. So, yeah, Richard.
Well, a while back, I saw a video where they were working on that actual
Uh-huh.
disorder, and what the doctor found is if you put like a mirror in between the patient
Mm-hmm.
and let the patient look at the mirror, so
Mhm.
they're missing their arm here,
Mhm.
they look at the mirror here of their right arm,
Mm-hmm.
and just think that you're moving the left and move your right too. Just that recognition is the brain just right, and then the brain's like, oh, it's not moving it, and then it realizes it's not there.
Yeah, so you can fool your brain. This whole idea of projection is fairly liquid. So I actually watched a YouTube video last week of something I've been teaching for years, but YouTube's always more fun. So they had this guy, this experimental subject, volunteer, and what they did was they put his arm under a table, and then they put a prosthetic arm in his shirt sleeve on top of the table, okay? So he's looking at a fake arm, and his real arm's under the table. And to complete the sensation in his brain of, yeah, that really, even though the guy knows, he's watched him do the whole thing, he knows that's not really his arm. But it's not long if they're tickling or otherwise manipulating the hand under the table. At the same time, they're doing the prosthetic above the table. They're doing the same movements to both. That before long, he says, wow, I feel like my arm's on top of the table. His brain learns projection that fast. So you can train people to use prostheses that way. And then in the YouTube video, the fun part is they stab it with a fork, which I think is kind of mean. So what does he do when they stab his hand with a fork? Even though he knows that's not his arm. So still, you train your brain fairly quickly, and that's why prosthetics are so useful. So like you said, the mirror image thing works if you've had the limb removed. I know of a case of a woman who had post-herpetic itch. Herpes is the shingles virus. Herpes zoster is the shingles virus. So if you've ever had chickenpox, you've seen the ads on TV. If you've ever had chickenpox, that virus is still inside you. Yes, it is. And shingles usually will outbreak when you're stressed. Y'all keep that in mind. And very often the first sign is out of the blue, you have diarrhea. Just boom. No fever, no nothing. Just, I gotta go. And then it's gone, and then 2-3 days later, you've got these little blisters from herpes, from shingles. We don't call it herpes, call it shingles. So if you've ever had a shingles outbreak, then you know it's pain. It's typically painful. It itches, but if you scratch it, it hurts. It's a burning pain. It's blisters, and then it goes away. Your immune system kicks in and drives it back into your nerve cells. Herpes zoster actually infects nerve cells. So fine, but in some people it hurts after that, and in some people it itches after that, and no amount of scratching will make it stop itching. So I know of a case of a woman who had shingles on her scalp, actually, and she would scratch until her scalp bled. I mean, she just couldn't stop herself from scratching. The blister's long gone, years before, and they had tried every therapy, every remedy they knew. And this was driving her insane. She couldn't sleep. It just itched nonstop. She actually scratched all the way to the bone till her fingers bled, and she couldn't. She'd be scratching in her sleep, could not stop herself. So she was desperate and willing to try anything. So as a last-ditch effort, they cut the nerves to the scalp so that itch sensations would no longer be carried. You know, surgery cures everything, right? It's still itch. nerves cut and it's still itch. Well, at that point, you know it's psychosomatic. There's some pathway in the brain, not in the skin, that's initiating this problem. So they did exactly what you said. They put her up in front of a mirror that inverted the image in a way that made it look like the left side of her body was the right side. And they went through some steps of therapy to make sure, poking this side and making it look like that side and so forth, till her brain was utterly convinced. And then they said, now scratch over here. And she did, and it stopped itching. Even though the itch was being generated on this side, just seeing that mirror image made it stop itching. It took more than one attempt. So she went back and went through therapy for several attempts. And finally, after getting down, scratching down to the bone, after getting the nerves cut, all it took was behavioral therapy. And she was done. She was cured. So projection is a sense that is absolutely crucial. But the idea of phantom pain then becomes problematic. So how would you treat phantom pain? It's really kind of difficult. But if you know where the nerve cell bodies are, and you know these are all those unipolar neurons, where are they? Unipolar neurons? They're in the dorsal root ganglia, right? So just because you had the receptors removed in the foot doesn't mean the cell bodies are dead. They're up in the dorsal root ganglia of the sacral plexus. So they may have to do, surgical ablation of those sensory nerve endings, and that, of course, is not a fine-tuned thing. So you may lose other senses as well. I have a friend who was a double amputee at the knee after an industrial accident, and his feet, the bottom of his feet would itch sometimes. So, when the bottom of your foot itches, there's nothing you can do except stop and scratch it. It's just that overwhelming a sensation. So he would leap out of his wheelchair and remove the socks that he wore over his stump and just sit there and just scratch the stump till he could find the right spot. And then he could scratch his foot, even though his foot had been long gone. So whatever works. So now you understand phantom senses. All right, so look back at your senses that you listed and tell me which one of those are chemical senses. Just mark them with a little check mark or whatever. Which ones are chemical senses? Okay, so what do you got? What do you know is a chemical sense? Smell? Taste? Anything else? What else is on your list? Vision, we know not. Hearing, equilibrium, we know not. What else you got on your list? Pressure, no. That's mechano. What else? You have your list right in front of you. What else you got? Touch is tactile. So not chemical. Remember, our mechano sensations included tactile and baro and ear. I know some of you had other things on your list, so just call them out. What? That's taste, so we got that one covered. Temperature, is that chemical? Can you taste hot and taste cold sensations from foods? Yeah, yeah. So hot, we said, was hot peppers, capsaicin. And cold, we said, was things like mint, menthol. We don't know exactly why those chemicals stimulate our thermoreceptors. We just know that they do. So those are chemical sensations that our brain interprets as heat and cold. Anything else? What else you got on your list? Look back at your list, because I gave us two categories. Chemical senses on the left, not chemical on the right. What else is on your list? Balance is with the ear. So hearing and balance over here. What else? There's one more you're missing. So look back through your list till you find it. Pain. Is pain a chemical sensation? Yes. You wouldn't think so, but it is. So when you think of pain sensation, what do you usually think of that causes pain sensation? Burning, cutting, squashing, like with a hammer. What are you doing to your cells in all those cases? You're rupturing the cells. And ruptured cells release chemicals. And some of those chemicals are able to stimulate pain sensation. Okay? So pain is specifically a chemical sense. All right? It's coming from pain chemical signals, coming from either damaged cells or destroyed cells. All right, so we're going to focus on the special senses from here until the end of this lecture. But before we do, I want to point out that we'll talk about other senses later. We're going to talk about the visceral senses when? Mostly next semester. Does anybody feel cheated by that, by not covering them? We'll cover them. We probably pretty much won't. Proprio. Actually, we kind of already covered those. In lab, you learned muscle spindles, right? There are also, we call them Golgi tendon organs. There's good old Dr. Golgi again. And we also have joint receptors. But we talk about proprioception in the context of our integration of pathways and motor control, which I didn't lecture on really. So it was more in the chapter summary for chapter 13, 14 than anything. Okay? And in general, proprioceptors are fine-tuning for motor control or for balance and equilibrium. So we may talk about them briefly when we talk about balance, but chances are we won't have time. All right, so we're talking from here on out just about the special senses. What were our special senses? The senses. Sight, which is vision. Hearing, which is auditory sense. Taste, which is gustation, or the gustatory sense. Smell, olfactory. And then balance, which is equilibrium. Okay, that's the 5 special senses. Remember, they're special because they're in the head. And they have fairly complex receptors. So of those, the simplest of them all are the chemical senses. And our chemical senses are olfaction and gustation. And they have chemoreceptors, which means that they have receptors in the plasma membrane of the cell that respond to specific chemicals and open particular channels, and that triggers a nerve impulse, and then that nerve impulse gets conducted to the appropriate part of the brain. All right, so what do you know about your sense of, let's see which one's first, your sense of smell. What do you know about it? Where is it? Where in the nose is it? You learn the parts of the nose. You learn the skeletal features of the nose anyway. So you know the perpendicular plate is the basis of the nasal septum. You know you have conchae, right? And then you have meatus. Did you learn the meatus? You learned the conchae anyway. The meatus are just the channels under the conchae. So what were the conchae? How did we distinguish them? Inferior? What else? Middle? What's above that? Superior. Thank you. Inferior, middle, and superior. Above the superior concha, in that very narrow part, up under the cribriform plate. That's the olfactory epithelium. So it's a very small patch of epithelial tissue. Okay? And how do you get there? Here's the diagram that shows it. If I'm trying to inhale smell all the way up to the olfactory nerves, it has to get pretty far up in the nose, right? So what happens if you have a clogged nose from allergies or a common cold? You can't breathe. What else can't you do? Can't smell. Okay, you've lost your sense of smell, we hope temporarily, although people do lose it permanently. All right, so you have to get the odorant molecules all the way up, and once you get them there, let's start at the bottom here in this picture. So the odorant molecules have to be dissolved in this layer, which is a sort of a seromucus fluid, and then they have to bind to these little, they look like cilia, don't they? We call them olfactory hairs. The olfactory hairs are really the dendrites of the neurons. And they're not sitting out in the air, but they are sitting out in this mucus layer. So of all the kinds of dendrites in your body, these actually project out through the living tissues and into the secretions over the surface of those tissues. They stick out further than any other dendrites. And your olfactory cells that they're part of, can you tell me what kind of neuron that is? It looks like this. So here's my little dendrites. That is a bipolar neuron. Those are the bipolar neurons of your olfactory epithelium. So each little olfactory hair is a dendrite, and each one of those dendrites has receptors. And each one of those receptors responds to a specific chemical or group of chemicals. So when the chemical bonds to its receptor, it's direct, which means the channel's open and sodium flows. And that's a positive excitatory post or a positive stimulatory impulse. And it gets the axon hillock right here to threshold. And once it hits threshold, it fires a nerve impulse. And that nerve impulse passes through the olfactory epithelium, through the openings in the cribriform plate. You remember looking at the cribriform plate, right? It's got a bunch of tiny little holes in it, doesn't it? That's what those holes are for. These nerves pass through it. It goes into the olfactory bulb, which is a part of the brain you learned. It sits right on top of the cribriform plates, right and left. And synapse is there. So your first order sensory neuron goes all the way from the mucus in your nose all the way to your brain. Pretty much a direct connection all the way to the brain. Does it make sense that you can get chemicals that are inhaled to your brain? Does it make sense that some psychoactive chemicals are snorted to get them to the brain? That's a pretty direct route. Okay? Does it also make sense that if you had a concussive injury to the front of your head, that you can crack the little cribriform plates and snip all those little nerves? So it's not uncommon for people who have an injury there. A friend that was in a car accident. It's kind of weird. She was crossing a road on campus. Y'all be careful on campus. We had a major accident here with a pedestrian and a car. And that means you have to be careful both when you're the pedestrian and when you're the one driving the car. Okay? So this friend of mine remembered halfway across the street that she'd left something she needed. So she turned right around in mid-street to go back the other way, back to her room. But the car that had been waiting for her to cross didn't realize she was going reverse direction right in the middle of the road. Who does that? So she got struck and lost her sense of smell for the rest of her life. So she got hit in such a way that it cracked the little cribriform plates. Okay? Other people have had it from getting beamed by a baseball or a softball. So be careful. Okay? The accident we had on campus was just last semester, and we've had 2 accidents in that lower parking lot between Pirtle and Jennico. And in both cases, I don't know the details of either one, but my sense of things is that on this campus, pedestrians are not looking both ways before they cross. I've seen students just assume that because they have the right of way, they have some force field around them that's going to keep me from hitting them. But I also know people drive too fast. What's the speed limit on campus? You ever looked at the signs to see what the speed limit is? It's 15. I go 10 because I've been driving here a long time and I know people don't watch. I would hate for one of you guys to be involved in cracking the cribriform plates of a student. That would be sad. So y'all be careful out there. Okay, there are other structures here. Sometimes, this is too much information. So I'm not going to ask you about the supporting cells or the basal cells or the little glandular cells or all that. But I do think you ought to have a sense of how lively this tissue is. Those are some of the few neurons of the body that routinely replace themselves. Now, we've taught for years neurons do not go through cell division. Because they're such complex cells, they really aren't set up to go through mitosis and divide again. And yet, these guys get replaced. Whether they get replaced by the basal cells, that's pretty likely. There's some stem cells there. I don't know. But I do think it's important for you to know that you do replace olfactory cells. And, of course, what happens when people get old? What happens always with aging? Sorry, everything slows down. So your sense of smell diminishes with age. This has a couple of different impacts for you if you're dealing with geriatric patients. One is their appetite's pretty much blah. Why would that be? Nothing smells good anymore, so nothing really appeals to them anymore. They tend to oversalt things. Why? To try to elicit some kind of flavor. And the little old ladies, bless their hearts, they wear too much perfume. Because they have to put it on until they can smell it. And they're not aware of odors in their environment. Their own body odors, okay, that's a fact. But also if gas were accumulating in their apartment or something like that. Okay, so be aware of that loss of sense of smell. All right, yeah, basal cells replacing. All right, that's all very interesting. Cranial nerve 1, we knew that, right? When it gets into the olfactory bulbs, where it goes next is kind of interesting. So we're going to follow it all the way to the brain. And we're going to see some convergence here. So you're going to have multiple cells that are carrying different impulses that converge in the olfactory tract or to the olfactory bulb. You already know from lab it goes from there to the olfactory tract, right? Mm-hmm. Okay, from the olfactory tract, where it goes from there, it diverges. And it diverges to, it says inferior frontal lobe. It projects to the primary olfactory cortex, which is in the temporal lobe. It projects to the hypothalamus and amygdala. It does not go to the thalamus. So remember earlier today I said the thalamus is your sensory filter for all of that sensory information. What's the temperature? How tight are your clothes? How loud is the air conditioner? Your thalamus filters all that out, but your thalamus does not filter olfactory. It's cranial nerve 1, and it attaches to the brain before the thalamus. And so, but you do get used to it, right? That's adaptation. So you have central adaptation and you have peripheral adaptation. Central adaptation means your brain is kind of weeding it out. Your thalamus does a lot of that. Peripheral adaptation means the sensory receptors themselves are getting used to the sensation. And so there's a combination of both. In the case of olfaction specifically, there's a combination of both. Okay? But that is the only sense that does not go through the thalamus. All right, so when you get used to smells, you ever notice how water, when you travel, tends to taste funny? And then you get used to it, right? And then you go home and your water at home tastes funny? Have you ever left your own domicile, whether it's a house or apartment or a dorm room, for an extended period, and when you come back, it has a smell that you didn't notice before? Yeah, especially if you have cats, let me just say. Get used to that. So the cerebral cortex gives you your conscious perception of smell. The hypothalamus controls your visceral reactions to smell. And the amygdala, which is a part of the brain we unfortunately don't have enough time to spend a lot of time on, the amygdala hooks your input into your emotions. So if particular smells evoke particular emotions, then you have your amygdala to either thank or blame, depending on whichever the case may be. So particularly in things like post-traumatic stress disorder, if smells trigger it, the amygdala response must have been particularly strong. And there are therapies now to retrain people with that, but there are other smells that might evoke pleasant sensations or memories of your childhood if you smell baking cookies or hot chocolate or whatever. Okay, so we're not going to worry about that yet. All right, so just quickly, we're not looking for a number here, but just an idea. Would you say dogs have about the same number of olfactory genes, more or fewer than humans? More. What makes you say that? It's not that they have a stronger sense of smell. It's not that they have a more sensitive sense of smell.
They use that instead of touch.
They do use that instead of touch, but what they have is the ability to detect odors that you don't detect. That's the point of having more genes. Each gene codes for a specific receptor region. The more genes you have, the more different receptors you can build. The more receptors you can build, the more different chemicals you can react to. So the correlation is between the number of genes and the number of chemicals you can detect. So some dogs are better than others. Name a breed of dog that is legendary for its sense of smell. That would be the bloodhound, of course. Duh. You just didn't think of that. Others as well, of course. Since the Human Genome Project was completed, y'all remember what that was, right? Take the entire 46 chromosomes of human, find out the AGCT code of every single chromosome. So, and build a humongous computer database. Actually, I had to invent the computing processes that would be able to read that. Well, of course, they didn't stop with humans. They've done chimpanzees, they've done laboratory mice and rats, they've done yeast, they've done radishes, all kinds of things. So what they found was that they can look for a gene that in humans is known for sense of smell. We know that we correlate this gene with an olfactory receptor. They have ways of telling that. And then they can go look for that same DNA sequence on the chromosomes of other animals, chimpanzees and laboratory mice and so forth. And what they found is that there are like 250 genes in laboratory mice that they were able to detect that correlates with a specific smell. And when they look for the mouse genes in the human genome, they find them, but they don't work. So we have the genes for detecting odors. Those genes have mutated in humans because as the ancestor of humans relied more and more on vision and less on olfaction, if those genes mutated, it didn't select us out. It was not enough harm to cause problems. So we have in our genome at least 250 genes for olfactory senses that are broken. It's broken, I can't smell that. But it does give you some sense of how well dogs and other animals can smell. And this should impact you if you have a dog. How many of you have dogs? Do you walk your dogs? I have to confess, I no longer walk my dogs. First of all, they're too lazy. Second of all, I have a huge fenced-in backyard. So I throw a ball and let them run that way. Why do dogs, they would much rather walk. Why do dogs want to go for a walk? They have to smell everything. And when you're walking for exercise, that's not when you walk your dog. You don't walk your dog well, you can walk them for exercise, but you better give them that long, slow walk if you're going to do it, because to them, that's like taking them to the movies. Okay? And if you tell them to hurry up, you're like saying, if you're reading a book, flip to the next chapter, we have to move on, and you can go sniff the next hydrant or the next tree, and you're making them quit before they're done reading that hydrant. So their sense of smell is that acute. They can tell a lot from that. And you're just really not being very nice to your dog. So I can't walk my dogs. I live out in a rural area with no leash laws. So if I ever tried to walk my dogs, all the neighbor dogs would come and it would just be a mess. Okay, so any questions about sense of smell? All right, gustation is our sense of taste, and this then hits our taste receptors, which are actually not the neurons themselves. The gustatory cells are separate receptor cells, and we call them taste receptors, and they're housed in the taste bud, which is not the same thing as the bumps on your tongue. So when you look at your tongue and you see those little bumps, those are called papillae. They're the lingual papillae, and you have 4 kinds: filiform, fungiform, valate, and foliate, and that's all very exciting, and that's what I call anatomical trivia. Okay? If they ever invent a Trivial Pursuit for anatomy students, you would want to know this. But I'm not going to test you specifically on the types of the lingual papillae. Okay? They do not all have receptors in them, so the ones that do, you need to know more about the taste buds than you do about the papillae. Okay? And, of course, why does taste decline after you hit 50, which apparently is old? I'm offended by that personally, but everything slows down, right? So you have replacement of these gustatory cells from the basal cells, and you have a reduction in that. Now, when you look at taste, let's skip that and move on. When you look at your sense of taste, what you see for the taste bud, it's kind of similar to the olfactory sense. You have little hairs, but these little hairs are not the dendrites of neurons. The neuron is way down here at the base. So the gustatory cell actually has a microvillus. It's not even a cilia, it's a microvillus. And that's where the receptors are. And the receptors respond to the basic tastes. And then the basal cells are there to regenerate them, and that's all just hunky-dory. If you get enough of a sensation, then it starts an impulse on the sensory neuron. And from our sensory neuron, we're going to go, this is the only special sense that doesn't have a dedicated cranial nerve. So let's stop and think about that. What are our dedicated cranial nerves for sensation? Sensory-only cranial nerves. Cranial nerves. We have olfactory nerves, right? What comes after that? Optic. So that is smell only, and optic is vision only. And what else? Oculomotor is not a sensory one, it's motor. So what are the other special senses? Remember, they all have to be in your head, so they're all going to run through cranial nerves. Trigeminal is not a sensory-only nerve. So yes, you're going in order, and that's good. And yes, it's sensory, but it's not only sensory. Vestibulocochlear, very good. So the vestibular nerve for balance, and the cochlear nerve for what? Hearing. Those are the only cranial nerves that are sensory only, okay? And of course, the vestibular and the cochlear are the 2 branches of the vestibulocochlear nerve, which is cranial nerve 8. That's it, 8. Okay? So back to our list. This is the gustation is the only one that's not sensory only. And it's the only one that doesn't have its own dedicated nerve. So the tongue has more going on than just sense of taste. In fact, sense of taste is relatively minor. You've got all those muscles you have to control, so you have to have motor control. You've got other sensations. You have sensations of pressure. You have sensations of temperature. You have pain sensations if you put something sharp in your mouth or you bite your tongue. So all of those things get mixed up. And so you have, in fact, 2 different cranial nerves, and remember these are right and left, 2 different cranial nerves that carry taste impulses. And you should have learned these already, because we did cranial nerves there on your test G today, if you haven't taken that test yet. Facial for the anterior 2/3, that's the primary one. Glossopharyngeal for the back, and in fact the vagus for the throat. Did you know you have taste receptors in your throat? We even found receptors for bitter in the lungs and the stomach. So bitter receptors respond to the presence of alkaloids, which are typically toxic. And they're bitter because what? What happens when you eat something bitter? What are you supposed to do when something bitter gets in your mouth? What would my 2-year-old grandson do with that? Spit it right out. And that's what you're supposed to do, because bitter means alkaloid, and alkaloid is toxic. Okay? So when we cultivate a taste for bitter, that's a very modern human cultural thing. Bitter back in the day meant poisonous. Spit it out. Okay? So you have them in the back of the tongue, those big valate papillae, the big ones. And then you have them in the throat, in the upper stomach, and even in the airways of the lungs. Okay, so where do we go from there? Our pathway takes us to the medulla. From the medulla, we're going to go to the thalamus because the thalamus is our sensory filter, and then it's going to relay taste to the primary gustatory cortex, which is in the insula, which we haven't talked about. So the insula is associated with the temporal lobe, and so it's part of the cortex. All right, in the medulla, there's other things going on. You've got some reflexes associated with not just the taste receptors, but just the receptors in your mouth that trigger salivation, secretions in the stomach. If it's bad, then that's going to trigger you to gag or vomit, and then, of course, your thalamic relay. So we'll learn all the reflexes associated with that next semester when we do the digestive system. All right, so there's your gustatory pathways. Any questions about those? Do you need to know the names of the nuclei in the brain? Does anybody feel cheated if we don't assign those? All right, basic taste sensations. This is not as clear-cut as we used to teach. We used to teach a map of the tongue. Sweet was on the tip, sour on the sides, bitter in the back. And bitter, yes, it is in the back, but the others apparently are so variable from one person to the next that you cannot say much about the others. But you do need to know the associations that your brain interprets with what chemicals we would associate with them. Sweet with sugars, also with artificial sweeteners. Salt with sodium and potassium. Sour with acids. Bitter, as I said, by alkaloids. And umami, my favorite. Umami in English is called savory. I like it better because it gives a sweet, salt, sour, and savory. And then bitter, which we won't get confused with. But there were American researchers trying to nail down these receptors, and there were these Japanese guys trying to nail down the receptors. Guess who won? Umami, Japanese. Okay. So that, though, is the receptor associated with certain amino acids. It gives you what we have a sense of as a meaty flavor. And in fact, it's mostly the amino acid glutamate, which we've talked about. Glutamate is in the flavor enhancer sold as Accent. And it just tends to make the food taste meatier somehow. Okay? You also have thermoreceptors. You have tactile receptors, so you can tell the difference between something that's crunchy and something that's saucy. You have water receptors. We know that you have water receptors. We don't know exactly how they work. Okay? All right. Next question. The gene for sweet receptors in cats is mutated. What effect does this have on the behavior of a cat? Kind of a dumb question, but talk about it. See what you think. So if you offered your cat something sweet, they don't know the difference. They flat can't taste it. The gene's mutated. It doesn't work. So they apparently are still producing what is sweet receptors. They just don't respond to sweets. So cats don't, they could take it or leave it. Okay, that was just kind of interesting. Did you also know this is something that you can use? Trivial fact, the sugar receptors, the sweet receptors of flies are in their feet. Why would that be? How do they explore their environment? They land on things and they walk them around. And as soon as they hit a little pool of sweet juice, whatever, it causes a reflex that makes their tongue extend. It's reflexive. It's just a strict spinal reflex. Well, they don't have a spine, but you get it. All right. Let's talk about projection and its association with taste and olfaction. So we know that your sense of taste is based pretty much on your sense of smell, okay? And you know if you get a cold, nothing tastes at all. Even though it's your sense of smell you lost, you can't taste anything. So I want you to explain that in terms of projection. So do you remember what projection is? Anybody need help with that? All right, so you explain to me why you lose your sense of taste when you have a cold. In terms of projection, put an answer down on your paper. I think a good start would be to say what projection means in this particular instance. Okay, so what's projection? Projection is the interpretation of the source of stimulus. So when you are eating, where's the food? In your mouth. So explain projection of taste, knowing that taste is a combination of olfaction and gustation. Okay? So explain how projection works when you don't have a cold and you can taste your food, and then explain why when you have a cold, you've lost the ability to taste, even though your mouth is working perfectly fine. Not really. All right, so how does projection normally work? Say you're eating a nice chocolate chip cookie with pecans, because I hate walnuts. What do you taste? Chocolate, what else? The pecans, I can taste them. And I can taste if it's walnuts. Can you tell if you made it with butter or margarine? Can you tell? Oh, yes, you can. And yes. Can you taste the sugar? That's 4 different things, right? And of course, to be able to tell the difference between pecans and walnuts, they have similar consistency, but they taste different. They do. Trust me, they do. Walnuts are nothing compared to pecans. Okay? Butter and margarine, similar consistency, they taste different. Okay? So now, what were our taste sensations? What did we have for taste sensations? Salt, sour, sugar, savory, bitter. That's it. So if I'm telling you I can tell the difference between pecans and walnuts or butter and margarine, am I depending on my sense of taste to do that? No. Because taste is sweet, sour, savory, salt, bitter. So where is my pecan versus walnut? It's not just in my memory, although it is there. It's in my sense of smell. Okay? So why do we call it a taste sensation if it's actually a smell? If I'm smelling flowers, I'm using my nose to inhale the aroma of a nice bouquet of roses. And I associate that smell with the airflow through my nose. And my brain projects that sensation back to my nose, no problem. But when I'm chewing a chocolate chip cookie, you know, if I walk in the kitchen and I smell them, I get that. I'm smelling it. But when I got it in my mouth and I'm chewing it, where does my brain project all of the sensations associated with that cookie? The sensations that are hitting my olfactory receptors, totally ignored by my brain. My brain knows where that cookie is. I've got temperature receptors, I have tactile receptors, and I have taste receptors that are all telling me the cookie's in the mouth. So that sensation of, is it pecan or walnut, is it butter or margarine, that sense of smell, but that's no other sense. There's no tactile stimulation of my nose. There's no airflow to my nose that's not also hitting my mouth. So my brain is tricked into thinking those sensations should be associated with the mouth. So when you project your sense of smell, you project those olfactory sensations, you project them back to the mouth because that's where the food is, and that's normal. So normally you project olfaction to the mouth while you are eating. Does that make sense? So what happens when you lose your sense of smell temporarily because you have a cold? Yeah, you know the food's still in your mouth, you're still eating it, but your nose is blocked. Your brain has not learned that trick of distinguishing, oh, that's olfaction that's messed up. And so your brain says there's not enough signals coming in to associate that with anything, so there's no pleasure. Does that make sense to everybody? Any questions about that? Okay. Next is going to be the eye and ear, and we're going to start that tomorrow. Okay? So make sure your name's on a sheet of paper. What do you have left to do today? You probably have my test to take, although a few of you took it before class. If you have lab this afternoon, focus while you're in lab on the lab stuff. And then what's the latest the testing center will let you in? 7. And if you get there when your watch says 6:59 and their clock says 7:01, no, you're not going to get in. So do not get there late. Get there early to make sure. Study over there so that you don't miss it. All right, see you tomorrow.